The Great Passenger Pigeon Comeback is Revive and Restore's flagship project that launched the organization's mission to bring biotechnologies to conservation in 2012. For 10 years, many partners helped slowly build foundational concepts for the program, and in 2022, we were able to launch full-scale research for genomics, genome editing and rewriting, and the reproductive biotechnologies needed to create a new generation of passenger pigeon-like birds that will enable their repopulation and restoration to the wild. The first thing we need is a closely related species in which to engineer the traits of the extinct passenger pigeon. The passenger pigeon was native to the eastern forests of North America, and its closest living relative, the band-tailed pigeon, occupies forests in the west. Band-tailed pigeons have a virtually identical diet to passenger pigeons, eating the western variants of woodland seeds and fruits that passenger pigeons ate. They are nearly the same size, and when breeding, they both lay one egg per clutch. While band-tailed pigeons could certainly thrive in eastern forests if introduced, they could never fill the niche of the passenger pigeons due to one huge difference: their social behavior. Passenger pigeons lived at extreme densities, always flocking and breeding in social groups, while band-tailed pigeons breed in territories with lots of space, rarely concentrating densely. We can't just engineer the bird's behavior alone. Differences in development, plumage, tail shape, and immune systems are all key traits to recreating ecologically functional passenger pigeons that will thrive. We need to decipher the genes underlying these traits in order to transform living band-tailed pigeons into passenger pigeon-like birds. Luckily, there are hundreds of passenger pigeon specimens in museums from which to sequence their genomes. By comparing multiple genomes of each species, we can figure out which differences matter for de-extinction. The second step is gene editing, which starts with culturing cells that can be used for future reproduction. In these cells, we can use CRISPR-Cas technologies to remove regions of the band-tailed pigeon genome and overwrite them with synthesized passenger pigeon sequences. Once all the necessary changes have been made, the cells are injected into a surrogate parent at an embryonic stage, where the cells mix with the migrating germ cells and integrate into the developing ovaries and testes. That egg is patched up, incubated, and the bird that hatches is called a germline chimera. When breeding a female and male germline chimera carrying our gene-edited cells, we will hatch a new form of passenger pigeon. This is where the biotechnology ends and the experienced work of conservationists takes over. Our plan is to have the germline chimeras breed continuously to rapidly produce large numbers of offspring. Those babies will be transferred to foster parents conditioned to breed in colonies like the historic passenger pigeons. They will grow with the visual and auditory stimuli of a flock of millions of birds with the use of mural paintings, decoys, and audio recordings, the same ways that seabird colonies have been restored for decades. When those babies grow up, they will breed and produce offspring of their own, in the same colony setting. Once there are enough birds to consider releasing to the wild, they will begin conditioning for release, living in large outdoor flights to be exposed to weather patterns, the elements, and the sights and sounds of other species. To replicate their nomadic movements, the birds will be cycled to different flights with the changing seasons until they are ready to begin free release. At that stage, the populations will be under long-term monitoring, supplemented yearly until they are self-sustaining, and one day, decades in the future, will be an independent wild species. We have 3 major areas of research underway currently to make all of this possible. A team at Rockefeller University has sequenced high-quality genomes of over a dozen band-tailed pigeons and the ancient DNA fragments of nearly as many passenger pigeons to identify the differences between the species. But to make sense of which genes are related to specific traits, we are looking to the genomes of other related species and pigeons that share at least one convergent trait with the passenger pigeon. Thanks to a partnership with a private conservation breeder in Qatar, we now have blood samples from nearly 20 species for our research. While the genomics analyses proceed, several teams are working to establish the cellular and reproductive techniques needed for germline transmission in pigeons. Starting with domestic pigeons, we aim to culture primordial germ cells, the proven cell type for this technology in other birds. We are also exploring alternatives to creating reproductive cells through embryonic stem cells and induced pluripotent stem cells. These technologies could fundamentally revolutionize conservation for living endangered birds. For genome editing, we need to prove we can make large-scale alterations to the pigeon genome to change behavior, plumage, and physiological traits. Researchers at New York University Langone Health and Linköping University, Sweden, are collaborating to use the chicken as a test model for de-extinction scaled genome rewriting. They plan to overwrite regions of the domestic chicken genome to alter the same classes of traits, behavior, plumage, and physiology, to transform the domestic chicken back to its original wild form, the red jungle fowl. This is a critical first step to proving de-extinction is possible. These 3 lines of research will converge in the late 2020s to allow us to begin recreating a new passenger pigeon, which we aim to hatch in the early 2030s. The road to restoring the birds to the wild will take decades, encompassing years of breeding, preconditioning, and then monitoring in the wild, affording time to plan and implement a safe and responsible program that benefits both people and the environment. Rewilding efforts like this need community and stakeholder involvement, which we at Revive and Restore are dedicated to engaging. With diverse public, tribal, and government stakeholders, new passenger pigeons can be restored in our lifetimes.